Welcome. Welcome. I'm gonna get started now. Feel free to keep popping up in the chat. It's so wonderful to see familiar names and hear that even though people are far from us, they can still participate in this format. So that's really wonderful to hear. So thank you all for joining tonight's presentation. I want to thank everyone for pre-registering and sharing your questions in that registration form. Our speakers have done an amazing job of integrating your questions throughout the presentation. We're getting started right now. Thank you for using the chat. So Dessert with Discussion may seem like an odd title for a program in these times and in this virtual format, but it's been an annual or biannual part of the or twice a year part of the Kellogg Biological Station's programming for over a decade now, and we just wanted it to keep going. So here we are, and we're hopeful that perhaps in a few months, maybe for Fall Dessert with Discussion, or perhaps for Spring 2022 discussion, we'll be together in person with desserts catered by our fabulous conference center. Just a little bit of Zoom etiquette in case you haven't spent all day on it like most of us have. Don't worry about it. Your mic's off, your camera's off because we're in a webinar. You'll just have video and mic for our moderators and presenters tonight. We are recording tonight's presentation and it will be available for you to share and watch again after the program. To ask questions tonight, please use the question and answer function. So you have two ways to interact with us. You have the Q&A and the chat. So the chat, Misty and I are moderating. You can ask us questions, but like logistical questions or if you're having trouble. But if you want to ask a question of the speakers, please use the Q&A to put that question in. And then if you want to interact in that Q&A, you can upvote by clicking the little thumbs up for a question that you want us to tackle. All right. A few introductions are in order. So I'm Kara Haas. I oversee community outreach at Kellogg Biological Station, along with my colleague, Misty Klatz. We will be moderating tonight's discussion Q&A in the chat for you, so let us know if we can help in any way. And our host this evening is the KBS director, Fred Jansen. And to keep things rolling along, I'm going to turn the mic over to Fred. Are you ready, Dr. Jansen?
Got it. Thank you very much, Kara, and I appreciate the opportunity to greet everybody again tonight. Really excited to be the new director of KBS. I've been on the job since last June of 2020. And this is my second opportunity to participate in this awesome Dessert with Discussion sort of series of events. And I think what we'll do is we'll start, importantly, I think, with a land acknowledgement. And that goes as follows. Michigan State University and the W.K. Kellogg Biological Station occupy the ancestral, traditional, and contemporary lands of the Anishinaabe, the Three Fires Confederacy of the Ojibwe, Odawa, and Potawatomi peoples that was seated in the 1819 Treaty of Saginaw. And we acknowledge that relationship. And I thought I'd start before handing it over to the really important part of tonight's presentation by giving you, for those who aren't familiar, a little bit of a rundown of what KBS is. Excuse me. It is Michigan State University's largest off-campus education and research center. It turns out it's also one of North America's premier inland field stations. And there are quite a few of them, but it's one of the very few that has year-round activities that go on in terms of collecting data and other kinds of activities on our facilities. KBS consists of diverse habitats and state-of-the-art excellent research facilities that again support resident faculty, students, visiting researchers, and again in a very unusual sense in a sort of a year-round rather than only a seasonal kind of a situation. So again, it makes KBS not only unique, relatively unique, but also a world-class kind of place, biological station. In terms of these facilities, they include 3,800 acres of research lands and other kinds of facilities. They've got roughly 150 people that are employed by KBS, and many students, faculty, and of course we have all sorts of other facilities that aren't necessarily research-based. And some of those are shown here. I think it's really important to get a sort of feel for those of you, again, who aren't familiar with the area in Southwest Michigan, kind of where KBS is and what it constitutes. So if you look at the little dark green inset in the lower left of the screen, you'll see a white star, and that shows in the southwest corner of the state where KBS is located near Kalamazoo. And then if you look at the rest of the map, you can see that it's situated with the yellow stars on Gull Lake, a very deep freshwater lake. And the research facilities are spread around there, and they include not just the long-term ecological research site, the Great Lakes Bioenergy Research Site, and so forth, but also some conservation sites, the bird sanctuary, and also a working sort of sustainable agriculture farm, the Farm and Dairy Center, and so forth. So it's really diverse, and again, world-class set of natural and managed ecosystems. And again, it's world-class. It's not just locally spectacular, it's also globally so. A lot of the work that's done focuses on 4 areas, primarily ecological, evolutionary, agricultural, and conservation biology, and usually a sort of integrated mix of questions that touch on those kinds of areas. And I think it's also important to let you know, if you aren't already familiar with it, that the kind of research that takes place is along a continuum from basic to applied research. And what we mean by this is basic is sort of that discovery-based, question-motivated kinds of research that helps make discoveries that we just really didn't know about before, relatively undirected, and all the way to applied research, which is the sort of thing where there's a specific question maybe directed at us from society, from the farming community, agricultural community, and so forth, where we're trying to answer a very specific, relatively, say, narrowly focused kinds of questions. And so everything in between. So KBS is all of that. And so with that sort of brief description of some of the things that we do at KBS and reasons for its existence, what I'd like to do is talk a little bit about our program tonight. Tonight we've invited 3 really top-notch scientists to share with us some of their work on soil health, which, as you may or may not know, and you will after tonight, is an important global indicator of environmental health. First, we have Dr. Sarah Evans. She's an associate professor at KBS. She's also affiliated with on the main campus of Michigan State University, the Department of Integrative Biology. At KBS, she runs a very vibrant lab, and that includes the other 2 presenters tonight, Dr. Jennifer Jones, who's a postdoctoral researcher, and Tayler Ulbrich, who's a doctoral candidate. I'll tell you briefly about Dr. Jennifer Jones. She is, again, a postdoctoral researcher, and that means she's already received her doctorate, so she got her PhD from the University of Illinois in Urbana-Champaign. And then Tayler is working toward her doctorate, and she's in the final year of her doctoral studies in Dr. Evans' lab. And what's really neat about this team who's going to speak to us tonight is that they offer an example of a sort of collaborative and innovative group that conducts cutting-edge scientific research. And I think we're going to be really excited, I certainly am, to learn a lot more about soil along with them tonight. And so with that introduction, I'm going to pass the mic to Dr. Jones to start tonight's presentation.
Awesome, thank you. And thank you everyone for being here. Kara put it in the chat, but there's over 90 people attending right now. So I'm gonna give a quick overview of the talk today. First, we're going to give brief introductions. Then we're going to define soil health. We'll talk a little bit about why soil life matters, with an emphasis on microbes, which is our expertise. And then we'll talk a little bit about what studies we're doing right now on soil and what we all can do to improve soil health. Slide. So my name is Jennifer Jones, and I grew up in Portland, Oregon, where I gained a love of nature. And I went to college to undergrad to study environmental policy. And while the more I learned about environmental policy, the more I got interested in the science behind those policies. So I started doing ecological research. Since then, I've done field research in Colorado, Florida, Panama, and now Michigan. And I'm really fascinated by how fungi and bacteria interact with plants and drive ecosystem processes. Taylor?
Hi, everyone. Thanks so much for being here. I'm excited to share some of what we're doing with you. My name is Tayler Ulbrich, and I'm a PhD candidate at the Kellogg Biological Station. And I'm originally from Des Moines, Iowa, where I studied at Grinnell College. And here's a picture of me on the left. When I was at Grinnell, I was really interested in learning about water quality and learned about how important our agricultural systems are for affecting our natural resources. After my bachelor's, I had the opportunity to study in Southern Africa, where I worked with my colleagues to interview Namibian farmers about how climate change is affecting their food availability and how they can farm their own land. So these two experiences really taught me that soil is a critical component to improving our agricultural systems and inspired me to do a PhD program at Michigan State, where I'm studying how soil microbes affect plants. So now you can find me doing things like running giant greenhouse experiments and looking at plant roots or taking soil cores while I'm trying to study how plants are interacting with this diverse array of soil microbes. And I'll pass it to Sarah.
Hi, my name is Sarah Evans. I'm the leader of the lab that Jennifer and Taylor are in, and we'll talk about what that means later in the talk. I'm originally from Alabama and started my interest in the underground by being fascinated by caves in the Southeast. So here's me with my sister kind of wondering what's inside that black hole. I have been concerned about environmental problems from a very early age, and I've always loved biology. So this kind of brought me naturally to a career in thinking about the biological aspects of sustainability. I really love studying different landscapes and traveling to see different ecosystems and comparing them. And I love that at KBS I can study these big global problems that we're facing using experiments in my own backyard. Slide. So I'm going to go ahead and take the first part. Return back to our roadmap real quick. First, we're going to take kind of a big picture view and just think about kind of why is soil important and what is soil health. Slide. So I want you to ask for a little audience participation here for a minute. If you could put in the chat, what's the first word you think of when I say soil? And if you don't mind, and there's an option in the chat to put it to all the attendees as well as the panelists. So it'd be great if other people could see your answers if you clicked the panelists and attendees, and just kind of enter what you think of when you think of soil. So I see smell and life, growing stuff, garden. Great, great. Root nodules. The earth, joy, alive. I love these. These are awesome. Cool. Those are great. I'm glad everyone had a different perspective. Bugs and worms. We're going to talk about that. Agriculture. We're going to talk about that. Microbes. Great. These are great. Okay. Can you go to the next slide? Keep going if you want. So I'm glad everyone, I feel like that shows we kind of have a multifaceted view of soil, which I'm thrilled about because that's kind of what got me into soil as well. So Wendell Berry said, the soil is the great connector of our lives, the source and destination of all. And I really like that soil can be this foundation for the most basic form of life, the bottom of the food chain that fuels the rest of our life on Earth, the plants. And also it literally forms the ground that we stand on. Others have described it as the thin, dark line. So it's like this thin crust that's only such a small part of our Earth physically, but it supports kind of all of life. Can you go to the next slide? But we didn't always have this sort of appreciation of soil. You know, there was a kind of a reckoning where we came to realize that our lives depend on soil sometime in the 1930s, when a combination of poor land use practices and agricultural degradation and many years of drought led to the Dust Bowl. So, you know, literally we were losing tons and tons of soil from the Midwest. And this was our livelihood, our support for growing food. And it sort of led to our nation kind of focusing on soil, and Franklin D. Roosevelt saying, the nation that destroys its soil destroys itself. So this event, a series of events, actually led to the formation of the Soil Conservation Service, which today is the NRCS. So it was sort of this, you know, actual policy recognition that we really depend on soil. And at the time, it was largely for the goal of growing food and supporting farmer livelihood on soil. So since then, it's sort of expanded. Slide. So now we know that soil supports a lot more diverse services than just food growth. It certainly grows food for us, which is obviously our basic need, but it also cleans water as the water trickles down through the soil to our streams and rivers. It holds great discoveries. This is where we get a lot of our medicines and antibiotics, from discoveries of unique genes and unique microbes in the soil. And it also regulates greenhouse gases. Next slide. So if you're wondering whether we could harness the power of that last one, that we can. So there's a tremendous opportunity right now for soils to help in our fight with climate change. This is because soils have this ability to tuck away the carbon dioxide that plants take up. So plants are photosynthesizing carbon dioxide, taking in carbon. And then that carbon can be sequestered by soil for a longer period of time. So this is sort of a natural sequestration of carbon. We're not injecting it, we're not using technology, it's just naturally what the soil does, this biosequestration. And it's the same with nitrogen, with another greenhouse gas, nitrous oxide. So there's kind of been some attention of this about this lately, is can we manage the soil to draw down the carbon emissions that we're putting into the air and help combat climate change. So Taylor and Jennifer talk about that a little later. Next slide, please. So what is soil health? You might have heard this term, you might not, but really informally, soil health, or a healthy soil, is means the soil is performing as many of those functions that I mentioned as possible. So, and these functions, especially that include beyond just growing food. So the USDA defines soil as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And there's a few key words that show kind of how our thinking is changing and evolving, because we used to think of soil fertility and soil health as really just a chemical thing. How much nutrients is in the soil? How much plants can you grow? But now, you know, we're seeing that word continued capacity. So we're really thinking longer term than just this year's yield or this year's plant growth. So to longer term sustainability. And we're also thinking of this as a living, breathing organism. So living ecosystem that sustains plants, animals, and humans. So we're connecting soil, not just to the plants, and not just kind of as a chemical fertilizer, but also to humans. Next slide. So if you're a gardener, you've probably literally gotten up close and personal with your soil, and maybe you already have this appreciation. And you're probably, if you're growing healthy plants in your garden, chances are your soils are also doing these other good services like sequestering carbon and cleaning water too. So most smaller land use practices are sort of encompassing some of those. But we're thinking, what could we do this on a large scale? Could we enhance some of these services and almost get back some of the carbon that we've lost from the Midwest in the last 50 years from harmful agricultural practices. So it would be a big impact and potentially save a lot of money, and that we would otherwise use cleaning the air and water by other means. Slide. So what do they recommend for soil health principles? So the USDA recommends 4 general principles for increasing the health of your soil. To keep the ground covered, so that's to prevent escaping of water and keep things warm and moist, minimize disturbance, grow diverse plants, and keep plants growing throughout the year. So I'll just mention now that the challenge is that this doesn't, or I guess I'll say the advantage of this new emphasis on kind of a holistic view of soil and soil health instead of just soil fertility and chemical fertility is that it's more holistic, and it acknowledges this complexity of soil that's kind of made up of chemical, physical, and biological factors. But on the flip side, it presents a challenge. It doesn't really allow you to have one practice that works for everyone, or a single score for soil health. So we're kind of working on that now. That's kind of our end goal, is to have some indicators and some specific recommendations for each region on what would lead to a healthier soil. So if we make sense of some of the science that underlies these services that are important for society, we may have some more general recommendations that are science-based for improving soil health. Next slide. So let's get into that science. That's what we're good at. So soil health can be divided into 3 interacting components, and I'll just introduce these before I hand it over to Tayler and Jennifer. Physical, chemical, and biological. Next slide. You may be most familiar with the chemical aspects of soil. So the chemical aspect is what gives the soil its nice, pretty colors, particularly iron gives the kind of that reddish tint to soil. But there's kind of a good and bad in terms of chemicals. Nutrients are good. There's not just fertilizer, there's also kind of micronutrients that plants need. And in the past, soil fertility was evaluated on its nutrient content. But you can see in the bottom left picture that there's also the bad. That's showing heavy metal contamination. The bottom right shows salt contamination, or salt, and a soil with high salt. And so we're increasingly, as a lot of you know, adding chemicals to our soils to monitor, or to control pests or weeds like glyphosate. And we really, we know that these are kind of reducing microorganisms populations, but we don't really know a lot about how harmful this is, in addition to being harmful to human health and kind of getting in our water. So potentially this is having adverse effects. Next slide. Cool. Yeah, so the physical aspects of soil, second component. The really cool thing about studying soil is that it opens your eyes to this whole new world. So when you look closely, soil isn't just a pile of sand. It's full of nooks and crannies, portals, tunnels, pores for microorganisms to live. And the healthier the soil, the more structure it has, and the more habitat it creates for microorganisms. So this is a little jumpy, but you can kind of see that this is what the soil looks like to a microorganisms, and maintaining that structure is part of soil health. Slide. Okay, the final aspect is the biological, and that's what we're going to focus on. In the end, the folks doing all the work in the soil are the organisms, is the biology. So soil is 100% alive and well, and as alive as any tropical forest or coral reef. But most of that life we can't see. So they have a whole food web happening, and the ones our lab primarily focuses on are fungi and bacteria. But you'll hear a little more about that from Tayler, and Jennifer. Next slide. Tayler.
All right, I'll take it from here. Thanks, Sarah. So we're going to zoom a little bit more into why the soil life matters now. Next slide. Thank you. So this is a really beautiful illustration that we found that illustrates this beautiful life underground. And so I just wanted to start us off with another quote from Wendell Berry, because I can't get enough of them. And it reads that if a healthy soil is full of death, it is also full of life. Worms, fungi, microorganisms of all kinds. Given only the health of the soil, nothing that dies is dead for very long. So I thought this was really beautiful and kind of alludes back to what Sarah was saying. The soil is both where we start and where we end. It is kind of the crux, it is the crux of life. So that's really inspiring and makes it really exciting to study. Next slide, please. So a handful of soil, I guess we've already told you the soil isn't dead, but it'd be difficult to tell that just by looking at it. Next slide. Because it actually contains an entire universe of life. Next slide. And one really crazy fact is that if you imagine every person that ever lived on Earth, generations and generations ago, that handful of soil contains more microorganisms than that many people. So that's a really incredible amount of microorganisms that we can't see, which again makes it really exciting to study and really unfathomable actually to think about those numbers. Next slide, please. So here is just a really quick overview of the soil food web, and I'll highlight a couple of the organisms that we think about. And so when we say microbes or microorganisms, we think about bacteria and fungi and protozoa and nematodes. So these are all those critters that you can't see with your naked eye, and we need microscopes or other technology to be able to see and study them. But what's really important to think about is that these are the basis of the food chain. So they're at the very bottom of the food chain. So that means that they support and sustain all life and organisms above them. So in addition to the other crazy functions we're going to tell you about and the roles they play, at the very least, they're the basis of our food chain and are important to think about. Next slide, I'll pass it to Jennifer.
Awesome. So Tayler and I are going to talk about a few examples of functions that microbes do in the soil. So the first one is that microbes can increase soil carbon. So here in the middle of the slide, we have a bunch of cartoon microbes, and they're decomposing plant litter and in exchange for carbon. And so microbes determine the rate of this decomposition, but they also determine what happens to that carbon once they get it. So microbes can choose to either respire that carbon, expelling it into the atmosphere as CO2, or they can put it into their microbial biomass or microbial tissue, which has the opportunity for that, that carbon to go into long-term storage in the soil. Next slide, please. One thing we'll talk about quite a lot today is the ability for microbes to alter plant growth. So here on the left is our plants with microbes, and on the right, they're plants without microbes, and they're not doing so well. So microbes are really important for keeping plants growing and healthy. Next slide. So one of my favorite research topics is studying plant drought tolerance. And understanding plant drought tolerance is so important because with climate change, we're expecting to have changes in precipitation that may lead to increased droughts or increased rain in Midwest and across the entire globe. So here are some examples of how microbes can help mediate plant drought tolerance. So first, microbes can actually retain water in the soil. They can delay soil drying by expelling compounds which retain water, thus increasing the water that's available for plants to use in drought. Microbes can also increase the production of plant stress tolerance compounds called osmolytes. And these compounds help make sure that the cells in plants retain their structure during water stress. Next slide. Fungi can also increase water access for plants. So here's a picture of arbuscular mycorrhizal fungi. And these mycorrhizal fungi have a symbiotic relationship with plant roots. And so through these fungal hyphae, a network of plant kind of fungal arms, the fungi can reach into the soil further than roots normally would and access water and nutrients that the plant couldn't access by its own. Taylor, over to you.
All right, thanks, Jennifer. So another cool function of the soil microbes that I would like to share with you is that soil bacteria can actually create the fertilizer that plants need. And so while we put sometimes tons and tons of fertilizers in our gardens and on our fields, bacteria actually have the potential to create this nitrogen that the plants need. They can create actually up to 70% of a plant's nitrogen needs if we create the right environment for them. So on the right is a diagram of a legume. So imagine a bean plant or a pea plant. And what happens is these little nodules or these little lumps on these plant roots, bacteria live in there. And those bacteria are able to take nitrogen from the air. So this is dinitrogen gas that makes up 78% of our atmosphere. But plants can't actually access that nitrogen on their own. And so that nitrogen seeps into the soil, and bacteria then that are living in these nodules transform that nitrogen into a form that plants can readily take up and use. And so this is a really incredible process, and in exchange for this really helpful nitrogen that the plant needs, it's giving these bacteria both a home, this little nodule, and carbohydrates or sugars as a food source. And so this is just one example of how these bacteria are providing fertilizer to these plants. Next slide, please. And here on the left, we have a picture of that nodule. So this is a pea plant, and that nodule is cut open. And so you can see that that's a really great habitat, really moist. It actually decreases the amount of oxygen in there, which is really ideal for this nitrogen fixation process, this process from going from dinitrogen gas to ammonia. And the picture on the right might seem a little surprising. So this is actually a corn plant. This is an ancient variety of corn from Mexico. And it was grown in soils that were really, really nutrient poor or didn't have much nitrogen. And what scientists found is that these plants don't have nodules, but this plant evolved a different way to interact with these beneficial nitrogen fixing microbes. And so this plant grew aerial roots, so roots above the ground, and it secretes this mucus that is the ideal environment and creates the ideal conditions for these nitrogen fixing bacteria to live. And so this was really groundbreaking that scientists are now realizing that it's not just these legumes that have, so our beans and our peas, that have these nodules and these nitrogen fixing bacteria, but other plants are interacting with what we call free living. So they don't need this nodule to live in. Maybe they live in this mucus, some live in soil. And studying how these plants are interacting with these special microbes that fix nitrogen is a really great way to boost our soil's fertility without adding inorganic fertilizers. Next slide, please. Now, one other example is how microbes can defend plants from pathogens. So home gardeners out there might recognize this picture. Here are some tomato seedlings that are dying. Their little stem is rotting and they're dying from dampening off. So that's caused by a fungal pathogen and is a pretty sad sight to see. But there are some bacteria that can actually help plants deal with this. Next slide, please. So Rhizoctonia solani is one fungus that causes this dampening off disease, and that's what's growing here in this dish. Next slide, please. But you can see here in this white line that scientists put Streptomyces bacteria, so that's a type of bacteria, and you can see that it's actually pushing away that fungi. And so these bacteria are creating, it's like a chemical warfare where they're being able to create these chemicals that are fighting off this fungi. And so even though we can't see them, this invisible competition is going on below ground. And that's really important because you can imagine if this plant were to attract these Streptomyces bacteria, it might help them be less susceptible to this bad pathogen. Next slide, please. And the other thing I wanted to mention is that Streptomyces are really incredible producers of antibiotics and antimicrobials, and they're actually one of the largest sources of our pharmaceuticals that scientists are able to use to isolate these special compounds. Next slide, please. And another key point I wanted to talk to you about was the Wood Wide Web. Some of you might have heard this in popular science news. So these are these mycorrhizal fungi. And actually 80% of land plants associate with mycorrhizal fungi. And they create these really, really dense networks of hyphae underneath the ground. And so these really dense networks are exchanging nutrients and water and signals. Just like our internet allows us to communicate, these hyphae are allowing these plants to communicate and exchange information. So one example of some signals that these plants might send is if one plant is being eaten by an insect, that plant can actually send chemical signals to its neighbor through this mycorrhizal hyphal network and say, hey, you need to be defending yourself because we have bad trouble coming. So that's a pretty neat thing. These hyphae are actually able to facilitate communication among plants, and I geek out about that a lot. Next slide, please. All right, so we've told you about some of the really amazing functions of healthy soils and microbes. So we have another opportunity for some participation. We would love to hear what benefit of soil health intrigues you most. So you'll see a poll pop up on your screen, and you can click all of the answers that appeal to you. And we'll see what the audience reports back to which soil health benefit they're most excited about.
All right, keep your votes rolling in. We're at over half of us have participated, so coming in faster and faster. I'll give you guys 5 more seconds to answer the poll. All right, I'm going to end the poll and share the results. Taylor, do you want to interpret?
All right, so it looks like everything received high remarks, but it seems like people are most excited about soil's ability to combat climate change. And I agree, that's a really incredible aspect of soil, and I think we're learning more and more every day about the potential. Next in line was support natural diversity, grow nutritious food, and clean water. So maybe these latter 2 ones were newer information, and we're hoping we can share some more about it. All right, thanks for participating. Next slide, please. All right, so next we're going to zoom into how can we study soil, and I'll pass it to Sarah.
Yeah, so next slide. At Kellogg Biological Station, my lab, the Evans lab, does experiments to learn about some of the things Taylor was talking about, the biology of soil health. And we're going to tell you a little bit about that. But first I wanted to say just how we function. What do I mean by a lab group? A lab group is simply a group of scientists that study the same topic. But these scientists are usually at all different career stages. So in this picture of our lab group, 2 summers ago at the bird sanctuary, we have postdoctoral scientists who have already gotten their PhD, and are doing sort of a residency or internship after that to get more training. We have undergraduates, we have graduate students, we have technicians. So we all are in kind of different career stages, so we're kind of can all learn from each other and share knowledge, share lab techniques, and also form a fun community where we're all interested and, like Taylor said, geeking out about the same thing. So while everyone might have a slightly different project, and you'll see, Taylor and Jennifer are interested in different things, but we're kind of all working towards the same question of how do microbes interact with soil and how do they enhance services. So next slide, and I'll hand it over.
All right, so I'm going to share a little bit about some of the things I'm thinking about in my PhD. And one of the main questions that I've been really fascinated about is how do plants communicate with these beneficial microbes? So here's a little diagram that I made, and it might look like a plant that's leaking things out of its roots. And that's what I was trying to get across. So those little water droplets are actually root exudates, at least that's what they're supposed to be. And so those are carbon-rich cocktails that have different chemicals and sugars and carbohydrates that plants actually use to interact with microbes. Next slide, please. Those are the microbes. So these plants are spitting out these root exudates as a way to both communicate and feed these microbes that they're interacting with in the soil. And just one thing I'll mention also is that all of the carbon in a plant, up to 40% of it, they push out below ground. And so that's really remarkable and really important also in thinking about soil's potential to hold these carbons. And part of it is through these root exudates that are carbons feeding microbes that then, as Jennifer mentioned, are turning over and contributing to the carbon pool. Next slide, please. So here's just a picture of a wheat seedling, and you can see this mucus and these root exudates dripping out of its tips. And I just thought that was a neat way to visualize beyond my infographic how these root exudates look. Next slide, please. So you might be thinking, that seems tough to study, and I agree. In the field, I think it's pretty much impossible. There's lots of overlapping roots, they're really dense, and I don't know how you could dig into one little root and study its root exudates and its microbes and really understand the mechanism that's driving how these things are interacting and communicating. Next slide, please. So in my PhD, we use something called rhizoboxes. And so basically what that is a box that has a front that allows plants' roots to grow along the surface, and you can see them growing the entire time. And so in these boxes, I'm growing 3 switchgrass plants, which is a native prairie plant. It's also important for bioenergy, a potential bioenergy crop. And I can see their roots, and that allows me to carefully dig out their roots. And here I put these syringe tubes on there to actually collect those root exudates that the plants are pushing out, so that then I can analyze them and study what is in those root exudates. What is it that these plants are spitting out to interact with and communicating with the microbes that are in the soil? So this was just one neat way that scientists scale down and try to make the soil system more simple, because it's really complicated in the field, and if we want to understand any mechanism, sometimes we have to break it down and understand the small pieces before we can understand the bigger picture. Next slide, please. Now it's to Jennifer.
Awesome. So we've talked a lot about the different functions that microbes and organisms in the soil can do. And so this is one of the questions that I'm really interested in. What do microbes do in soil? Next slide. And so one technique that I use to study what microbes do in soil is called metagenomics. In metagenomics, we take a sample of soil, we extract and sequence all of the DNA in that soil, and then we get data that looks like these A's, T's, G's, and C's on the left, and so on the right. And so we use this DNA data to identify genes that will tell us the potential function of all of the microbes in the soil. And so this is really important or really exciting to do, because in one sample we can study all of the organisms and all of their functions in that one sample of soil that we took. And so it's taking a kind of different approach from what Tayler is doing, which is going out in the field and taking a single sample and seeing what is everything that can happen in that soil. Next slide. So in the Evans lab, we also do research that's related to farming. And so another question that I'm interested in is, how do soil microbes increase drought tolerance in crops? And so I've examined this in both corn and soybean. And I've looked at this from a perspective of both how do we improve soil characteristics for improved microbial communities and their function in soils, and also how can we add microbes to the soil in order to increase plant growth during drought. And so that's what I'm doing on the left here, is adding microbes to the soil. And when we do this in the field, in order to study drought on plants, we use what we call rainout shelters, which is this picture on the right. Basically, it's a structure with a clear roof and open air sides, so that we can block the rainfall while not changing the temperature and wind flow under the shelter. Taylor, back to you.
All right, so it might be clear by now that soil microbes and healthy soils hold amazing potential for our world and for the production of food. And so it's not surprising that this has caught on in the agricultural world. And so these are just clippings of a couple articles that have come out in the past few years that show how excited agricultural industries and farmers are about the role of microbes in their agricultural systems. So one of my favorites is From Flask to Field, How Tiny Microbes Are Revolutionizing Big Agriculture. And this has really excited me because we're in the labs studying how these microbes are doing it, and farmers are in the field interacting with the soil every single day. And so I really wanted to know, what do farmers know about soil microbes, and what do they know about soil health, and how can we use both of our knowledges to better understand the potential for these microbes to improve agricultural sustainability. Next slide, please. So for one of my dissertation chapters, I'm working with Michigan farmers and asking them what they think about soil health and microbes. So this is a picture not from my research, but from a round table at the Kellogg Biological Station. There's a lot of work at the Kellogg Biological Station trying to connect our research to what farmers are doing. And my research is just one more aspect of that, trying to dig into this aspect of soil health, which can seem like a big nebulous box, and trying to break it down to understand what farmers are thinking so that we know as scientists that we're studying the right things and communicating it correctly. Next slide, please. And now we're to Jennifer. Sorry.
So what is our role in improving soil health? Next slide, please. So this is a zoomed in picture of soil. You can see a plant root here and some different soil particles and some hyphae that's connecting everything. And so it's important to notice that, to understand that this is the home for a lot of organisms: bacteria, fungi, nematodes, other worms, and insects, other invertebrates. And so it's important to care for soil because it's home for so many organisms. Next slide. So one question that we got from the audience is, should I add microbes to my garden? Is that going to improve soil health? Is that going to improve carbon sequestration or improve plant growth? And so here's a picture of fungal spores, which can be used for helping plants grow, and also a picture of some plants that are doing way better when they have microbes than when they don't have microbes. And so it's clear that microbes have this great power to increase plant growth and do so many important ecosystem processes. But it's also important to know that microbes are kind of finicky to work with, and it's very difficult to get, when you're adding microbes, it's difficult to have them establish and grow in that soil. And it's difficult to have their function match up with the function that you want, either carbon storage or increased plant growth. And so we recommend focusing on making a good, healthy environment for microbes in your soil. And so if you make a good environment for microbes, then hopefully they'll be happy and do good things for your plants as well. Next slide, please. Okay, so outside of your garden, what can you do to support healthy soils? Well, first, if you don't have one already, I know a lot of you have gardens already, you can start one. And as you're gardening, make sure to remember that the soil that you're working with is home to so many organisms, and make sure that you use healthy practices with that. Also, you can compost. You can start a compost. You can even use the compost in your garden, and that's a really great practice for soil health. The other thing that you can do outside of from your backyard is to know where your food comes from. So if you buy food at a farmer's market, you can talk to the farmers that you're buying from and ask if they do any soil health practices on their fields. And lastly, just continue learning. So there are a lot of great resources. I think we'll send a few out tomorrow, but this is one, Kiss the Ground. It's a documentary on the potential for soil health to mitigate climate change. Next slide. And thank you so much for coming and participating on Zoom.
Great job.
Thank-
Yeah, go ahead, Fred.
Oh, I was just going to say thank you all very much. That's just incredibly educational and super interesting. I certainly know my longstanding history and relationship with soils is pretty old-fashioned. And working with my grandfather, I used to think of it as dirt, but not alive. And this is just, you know, just a spectacular sort of perspective on it. So I hope the rest of the audience gained as much as I did from this glimpse of your underground research world.
Yeah, at this point, we'll open it up for questions. Please feel free to use the Q&A or the chat box for questions. We did have one come in from Natalie. Does the growing of native plants help soil more with the development of microbes than other plants?
I can take that question. So that is something that new studies are coming out every year on that exact question, but I would say in general, yes. Planting native plants generally helps bolster native microbes because of just historic associations. Those are like more likely to have kind of mutualist or kind of good partners in the native microbes. So that's going to kind of amp up those native microbes. In addition, you know, a lot of native plants are just good at kind of placing, you know, carbon and other nutrients below ground. Other questions?
That's my unmute. We have some great questions. Next one is, are there communities of microbes that seem to specialize in disturbed environments and work well with unstable plant communities? Are some microbe specialists and some generalists?
I can take one.
Yeah,
Oh.
go ahead.
Yeah, it's a great question. And overall, yes. So one thing that I guess, so the first response is yes, microbes are specialists. And one thing that we're finding is that microbes adapt to climate change. So just like we think of plants and animals maybe adapting and changing their traits in response to climates, microbes do the same thing. So one really amazing study that was actually done at Kellogg Biological Station found that soils that were preconditioned to drought actually had a better capacity and ability to help plants grow under drought. And so what that's suggesting to us is that there are microbes that gain certain traits under stressful conditions. And in many cases, those are the microbes that are actually better at helping plants under stress. The same thing goes for thinking about if you're moving one plant from one area to another area in terms of like large geographic distribution, it might not do as well in a new area because it doesn't have the specialized microbes that it needs. And now some plants are more dependent on specialized microbes, but I would say overall that is one thing that we should be thinking about. And I think as scientists are trying to develop and think about microbes that can improve agriculture, we are thinking about what are the traits of these microbes that can be best used. Great question.
The next question you might tackle too, Tayler. It's a bit about farmers' attitudes towards microbes and are they adopting these practices? If not, like how that conversation's going.
That's a good question. So I'm just starting these conversations with farmers, but overall, I would say it's a very split camp. There are some farmers that are really bought in already and they're really excited and are doing these practices. And there are some farmers that are more hesitant. And what's important to remember is that there are lots of things that go into a farmer being hesitant. One thing is that it's their livelihood. And so making a big risky change, some of these practices are costly or they might make mistakes and not make as much money the next year if they do this. So there are reasons. But I think farmers generally, at least about soil health, are getting more excited. And so I've called 20 farmers in Southwest Michigan to ask to these interviews and say that I'm specifically interested in talking with them about soil health. And every single farmer has been excited. And so I think that shows that farmers are excited to learn about soil health. they're excited to learn how they can do it. And I think that the government and scientists need to help support farmers to help them make the transition to these healthy practices more sustainable and easier for them. I hope that answers your question.
Thanks, Taylor. I think that was great. All right, I love this one from Steve Allen. How can you tell if your microbes are happy? Happy microbes.
So that's a question we get a lot from farmers, because they want their microbes to be happy, and they want an indicator that they're happy. You know, they want like to see a happy face. So I usually say, like, a healthy home makes the microbes happy. So those principles of soil health, where you're keeping the ground covered, you're, planting things year round, you're just minimizing disturbance to the soil, that's going to keep your microbes happy. But it's actually a very profound question, because in our field, one big thing we're chasing after is indicators of soil health. So basically that happy face for the microbes. And you want an indicator that's cheap and tells you something quickly, but also is accurate. So sometimes we have these very complex scientific methods that can tell us what our microbes are happy, but maybe they're not very practical. So we really haven't found a good, simple indicator that can, that's widely used and practical, that can just be like a thermometer on, are my microbes happy? So that's still out there, and that would be really useful. But, we have some things that we can get at using that. And we do know what practices generally make microbes happy. So we can just encourage those.
And so I guess one thing I'll add is a little bit more specificity. Like, we can measure, like, if there's carbon cycling going on, so we can measure if the microbes are active. We can measure if they're processing nitrogen, so that would be an indication that they're happy. But other than that, there's a lot of techniques that don't even, there's so much we don't know about microbes. There are a lot of techniques that we use regularly when we don't even know if the microbes are alive or not, with the results that we get. So there's so much to explore.
Thank you. Anna wants to know, what did we think of the science in Kiss the Ground, and did they do a good job?
Taylor, do you want to take that question?
I watched Kiss the Ground twice now because I really loved it and thought it was inspiring. I think overall the science is sound, but I think that they're a little bit extreme with some of their statements. So one that stuck out to me is they said that if you put herbicides on your field and all of a sudden you will have no microbes. And that's not true. I think you would be hard pressed to find any place that has no microbes. You might not have the microbes that you want, but you're going to have microbes there. So I would say overall, I thought that the science was sound, but just take it with a grain of salt that they had a very clear message, and that some of their ways of emphasizing it might have been a little bit strong.
Thanks, Taylor. We did have another question about, do herbicides damage microbes? So I think we've touched on that a bit, but I know from talking with you guys, this is an area that we're trying to answer that question, right?
Yeah, so I know some studies that show that herbicides inhibit growth of microbes, kind of like herbicides generally are proving to be bad for most organisms. And, they get in and disrupt cells. They are inhibitors of kind of reproducing of cells. So that's not good for biology. So I think that, this study was on fungi. So I think they're bad, but generally which species they target or what the implications of that aren't really known yet. I think it's known that they reduce the diversity, they kill some microbes, but it's sort of not known how big of a problem that is for microbes. Do other people know further than that?
I was just going to chime in that I don't know the exact science on this, but I also have read articles that are thinking about the lifetime of that pesticide or herbicide. So that differs a lot for different products, how long, like I think they call it I'm blanking on even the time, but how long that product will stay in the soil. And so I think that's something also to consider, just in how long of an effect it'll have on the soil community, is what is kind of the residence time, or how long will that product stay in the ground.
So this also relates to a question on other contaminants like PBBs or PFASs. And, you know, generally it is harmful to microbes, but I think some of what you see more research on is whether there are microbes that can degrade these substances. So that's one reason why these contaminants are so dangerous, is that because they're manufactured, and you know, a lot of them are like plastic components of plastics, and they're not like occurring in nature. So usually there's not a microbe out there that it's like their job to degrade it or to eat it. So they might kind of be able to break it apart and degrade it, some components, but there is some active research on kind of how we can either engineer microbes with gene manipulation, or kind of like Jennifer was talking about, like add microbes that may not be there already, but they may exist, to degrade some of these substances that stick around for so long. Because I think right now, honestly, I think they probably do harm microbes, but I think the bigger priority is their effect on human health. So we're looking at, you know, how long do they persist in the soil, and how do they get into our drinking water, and can microbes sort of interrupt that problematic cycle. So I think probably there'll be some more research on how it could impact microbes in the future.
Thank you. So I think some of the last rounds of questions here, and you've touched on this probably, and it's hard for me to listen and moderate these questions, but so I apologize if I missed it. But a couple questions about like a minimum level of microbial diversity. And I think hand in hand with that is, how do I measure that? Randall shared that my garden has 20 years with no pesticides, herbicides, and minimum tillage in the last 5 years at no-till. Can I assume the healthy community of bacteria, fungi are already there? How can I tell?
I would say yes, but I mean, I think you can, you know, this would be great. I would love, and people ask this a lot, it's like, I would love for there to be some indicator of like we talked about, the happy face. But generally, farmers at least know kind of like, they have these very sensory characteristics of whether their soil is healthy. And that if you have a healthy soil, there's no way you can't have healthy microbes, because they're the ones making the healthy soil. So you kind of have to have that first. So if you have that dark, you know, organic, rich soil that is like, has a lot of structure and, you know, is really weighty and full of kind of carbon and earthworms and substances and life, then that means you have happy microbes, and that they're doing their best work of like building nice soil. As far as a minimum amount, that is a really interesting scientific question, because we know that a lot of microbes do the same jobs in soils, so we don't necessarily need all of them, but we, you know, there is a minimum, obviously. And I think really it's like, it's really hard to identify where that like synergy occurs when you have everyone working together, working up the food chain, trophic cascades with earthworms and with nematodes. And, you know, you have this like synergistic effect where you get, you know, a healthy soil times 10. So that's kind of what you want, is like this ecosystem that's just functioning. And maybe you can get by, like it's still going to be soil and still be making nitrogen with a minimum amount, but you won't be like at your max potential. But yeah, I mean, that's kind of an outstanding question. And maybe Tayler or Jennifer have ideas about that.
Yeah, so I think something else I would add to this is that I think that there's the evidence that there is a minimum level of microbial species may come from like restoration projects where those are kind of some instances where adding microbes to the soil in the field can see some really good increases in plant growth and plant establishment and restoration projects. But generally, I think that there are a lot of microbes in the soil. So there's some really cool research coming out now, and some of which that's part of the Evans lab too, is that microbes can disperse pretty far distances. So if you plant some plants and have some soil that's had plants for a while, the microbes will probably come. They'll probably be able to make it there. Does that make sense to everyone?
I think that's great. And I just had one other thing I wanted to chime in with, is that an interaction that I had with a farmer who had 13 species of cover crops on his farm for a really long time. So Ron, sounds similar to your garden where you're just like doing really all you can. And he told me his livestock on his farm were his microbes. And one thing that he noticed is he didn't have to use herbicides at all anymore, because he didn't have issues with pathogens or pests because his soil was so healthy that that wasn't an issue. And so even though you can't look at directly who the microbes are and if they're happy, you might start to see functions that are happening in your garden that are proving that your soil is healthy. You might not have as many pathogens. Your plants might be more tolerant to drought, things like that, that are kind of just like a check for you personally that proves like you're doing a great job and really creating this healthy environment.
So there's several questions around year-round plantings, and Michigan having a very cold winter, and is that possible? So I think some of them are on the scale of cover crops and ag, but some of the questions are like, should I be planting something in my backyard that can grow year-round? So if you guys can talk about that a little bit.
So I just put a typed in answer. I don't know if everyone can see it when I answer it on the typing, but anything perennial that's going to save its root system for the next year, even if it's not growing above ground, like that's good, because it's kind of saving that soil structure and that below ground roots that you can't see for the next year when things aren't growing in the winter. So beyond that, I don't know, maybe Taylor or Jennifer have recommendations for keeping things in the ground in the winter.
I know that I thought about this in my own garden a lot. And so this year we actually did our first cover crops in our garden. So we had some clover and I think we had rye out there. And so we tried to get just a blanket of these crops that would still maintain their biomass and still be present throughout the winter, even though they weren't necessarily alive. And so they're keeping that root in the ground. One of the challenges with cover crops is you don't want them to get too big, and that might mean you have to till up your soil a lot or apply herbicides to get rid of them so you can plant your garden back in. But I think that can be a good practice, and you have to play with it in your own garden to see how you can figure that out. Another way is you can use mulch. So we'll put down leaves or cardboard, even just like to cover that soil. So that's not as good because it's not this root that's in there that's pushing out this carbon. But those are some things that I've done in my own garden to keep that living root in there.
Thanks. Next question's about the microbial community in no-till versus traditional farming.
Yeah, I would say there's pretty good consensus now that no-till does increase soil health. It's not always the case. Like there are areas or farmers, or if you're trying to do a different practice as well to maximize soil health, that you maybe can't do no-till as well. There's always kind of exceptions, but in general, it really just allows, especially those fungal hyphae that are kind of like roots, to grow through the soil. And every time they're tilled, they're kind of broken up, just kind of sliced up, and a lot of that carbon in the soil is mixed up and accessible to microbes, and it goes right back into the air, which is bad for climate, but also bad for the soil, because that is kind of what keeps a lot of the nutrients and stuff in soil. So I think that's a practice that's over the last decade, really some of the science has like shown that it can really benefit in most areas, not all.
And one thing that we know from KBS research is that no-till can increase plant growth during drought. And so while we don't quite understand all the mechanisms related to the microbes in the soil that might be related to that, like increased drought tolerance with no-till, we're investigating that and further, and we see an effect.
Thank you. Doing good on questions. This one is, do manufacturers of fertilizers and pesticides resist efforts to reduce the use of their products? And is this bad for the future of sustainable farming?
Sarah, do you want to say something about that, or?
I don't know the answer, but I'm glad you're thinking about capitalism.
Yeah, I'm glad you're thinking about it too. And I don't have an exact answer. Probably have my own biased personal answer, but scientifically I don't necessarily have one. But there is one example that I'd like to share that's related to this. And so there are a lot of ag companies coming out with microbial products called biologicals, and they advertise these as a way to reduce fertilizers eventually because you're putting the microbes on to do the nutrients instead. And I actually had an interesting interaction with a dealer for some of these biological products and asked if they thought eventually they would stop selling the nitrogen fertilizers in lieu of these biologicals, and they very plainly pointed out that they still make money through nitrogen fertilizers. So I think that the economics get tricky behind it, but I am hopeful that microbial and more sustainable products will become on the market, and hopefully if they gain more traction in the market in terms of farmers are wanting to buy that, then maybe they would gain more traction over the less sustainable practices and inorganic products. Yep. That's my thought on that.
That's a really good question, though. I don't know if we know that. I wouldn't be surprised if there is a lobby for that, but not explicitly, I guess. They're not like anti-soil health vocally, but I think it does take a lot of convincing for farmers to reduce inputs like from fertilizers, just because it's so ingrained in the practices to just always fertilize, and it's also very heavily subsidized, the cost of fertilizer. It's very costly to make fertilizer because it's an energetically intensive process, but it's subsidized so that it can be cheaper. So I think that's kind of a condoning of that practice, which is not inherently bad. We couldn't really live without fertilizer and produce the amount of food we have now, but it does sort of beg the question of like, which one should we allow first? How can we see the max potential of the soil to provide its own fertilizer without, if we're kind of continuing to add it?
Thank you for those thoughtful answers. This question is, how helpful are static aerobic composting systems to reintroduce microbial diversity into soils where diversity has been lost due to tillage? And if you guys know what a static aerobic composting system is, will you enlighten me? That's a new term for me.
I don't know what that is, except for a large, like industrial scale composting operation. Does anyone else know?
No, I don't have that much experience with compost.
I don't either. I know that aerobic means that it would be getting air as opposed to anaerobic, which is good. It means that you're going to be keeping it active. And the compost can hold lots of microbial diversity. But as far as that question, I'm also not.
Yeah, so I guess I'll comment on, just assuming it's a large scale compost. I think about this a lot in my compost. You all kinds of stuff are in your compost, and then you put it on your garden, and that's clearly an introduction of different microbes. But this is kind of why you don't put like meat or waste or something like in your compost, because there can be pathogens or bad bacteria that like are not healthy for us. So that's kind of why you put natural products, because those things do go into your garden and they can harm your microbes and harm your food quality. And so I think in general, you're using those microbes to kind of create fertilizer and create good soil that you're going to then add onto your garden. But yeah, there probably is some research about kind of how those colonize and affect the garden. But in general, I think if you're using good practices for compost, and some of those like large scale aerated compost probably do, then they're most likely the benefits are going to outweigh the bad.
And our resident compost expert, Andy Fogel, came to the rescue.
Oh, thank you, Andy.
Yeah.
Oh, formed and not turned. Okay.
Yeah. So that is interesting that it's not turned, I'm guessing, because like we were talking about soil tillage, that kills some groups of microbes, right? So
yeah,
a compost pile that is layered to have air movement but not be tilled might generate more microbes. That sounds like a undergraduate research project to me.
yeah.
For some summer to do some different compost pile studies.
Yeah, but if people compost, I mean, that's an awesome example of microbes in action, because and just if you get really into it, you're thinking about which foods or which, how much leaf material do you put, or grass clippings or foods. And those are recommendations are all based on what microbes like to eat and what their bodies are made of. So you're basically trying to please your microbes and in order for them to make good compost.
Great. Liana has put an example of the Johnson-Su system in the chat too. So we'll all have to check that out. Thank you.
Thanks for educating us.
Yeah. Awesome. Couple more questions. Ron wants to know, are we seeing negative issues with invasive microbes similar to invasive plants or animals?
I love this question. I think this is a great question. And maybe Sarah and Taylor have different thoughts on it, but I just think we don't know, and it's really difficult to study that because we can't see microbes in the same way that we can see a plant. So we really just don't know. We've only had kind of this DNA analysis for 20 years now, 15, 20 years now, and that we can use like on soils at that scale. And before that, we were using Petri dishes, and it's very, very difficult to identify what a microbe is and where it is and where it isn't. And so, so it's a very difficult question to answer.
Thank you.
Yeah, I think the most obvious example or the easiest to track is pathogens, so that's because we pay attention to pathogens right away. But if there's like an outbreak of something that wasn't in a crop field before, or an outbreak of something like COVID-19, that is an invasion of a microbe, and that wasn't there before, and it might capitalize on not having competitors or not having, or maybe disturbed area that it can kind of take advantage of. And we definitely see this, but I think it's hard for us to identify when there's an invasion that we need to pay attention to, because there's microbes like blowing around in the wind and being traveling on animals all the time, and so there's always invasions. And maybe we don't, in the same way with plants, we don't really see like, oh, that's a problem. But so I think it's just a matter of learning like which ones we should pay attention to.
Yeah, I think that's a really good point. Like we see it when it's a plant pathogen, but we might not see it when it, inhibits soil carbon storage or something like that we can't see with our eyes. Taylor, do you want to?
I was just going to chime in that that's a really interesting question also in terms of thinking about these products of adding microbes to our lawns or to our agricultural fields. because that's one thing that people are considering is if we have this just microbe that we grew in the lab or we isolated from this one specific place and we market it to put it everywhere, is it going to be good everywhere? Could it be bad in some places? I think that's one thing that will really become an issue of a topic of discussion when more microbial products start to be marketed and sold globally, because it's really something we'll have to consider.
I think two questions to wrap up. One is really specific. How beneficial is poultry manure? And yeah, that's the question. How beneficial is poultry manure? And I think maybe in the chat too, there were a couple other questions about manures or and obviously certain types of composts were talked about. So I think we can't speak to poultry manure possibly.
I don't know the answer to that. Someone else might. I mean, I don't know how it compares to other like cow manure. If there's concerns, especially like if there's antibiotics, that is concern, just like it is for cow manure, can be released into the environment and cause antibiotic resistance. But I think otherwise it's pretty safe. I'm not sure.
It's stinky from my experience. All right, my last question is, does diversity equal happy?
Yeah, I was hoping you'd take that question. So I'll let other people chime in, but I would say often, but not necessarily always. So that's one metric we might use to kind of indicate a healthy soil. And it can kind of show that you're, maybe planting diverse plants or that there's a whole soil food web out there. But if you can have a diverse system of pathogens as well. So can't really be the only metric. That's a very big question.
It's a big question.
I would
All right.
totally agree.
Go ahead, Taylor.
What I was going to say is that it gets one of the other reasons these soil health comparisons get really tricky is because every soil type, every location has a different capacity to hold a different amount of microbes potentially. Just every soil has its own potential. So every soil isn't going to be able to get to the same level of soil health. And so that's also important in thinking about, if this soil has way more diverse microbial community than another one, that doesn't necessarily mean it's more healthy than another one. It's just the microbes that are there, but it's really critical to look at those functions, and that technique metagenomics that Jennifer told us about is one really neat way we're trying to dig into the diversity of the functions of the microbes, not just who they are. So we're hoping that that'll help us get a little bit more in detail about that.
Yeah, and so there are whether adding diversity has a positive effect could also likely differs between the function that you're interested in looking at, like Taylor was kind of getting at. So there are some functions that a lot of microbes do, like decomposition, and then there are some functions that only a few microbes do. And so if you add those microbes in, then that increases that add the diversity in the soil, and that increases that function. But yeah, it's complicated and a little controversial too.
Thank you. Great discussion. Thank you to everybody who put a question or thought in the chat or the Q&A parts of Zoom. We really appreciate that participation. I will send you an evaluation tomorrow in my follow-up email, and would love your feedback on this program and what you would like to learn more about. We hope that you'll keep in touch with us if this is your first interaction or your 999th interaction with us at KBS. We value that relationship with you, and we hope that you'll engage with us in ways that work for you. So join our mailing list if you're not on it already. Visit our website to do that. And tomorrow is Give Green Day, a day of development and fundraising across Michigan State University. We have a special project that our longtime donor, Dr. Dave Dvorak and Carol Peterson, are matching up to 10 grand to put some cameras at the bird sanctuary. So if you want to help us make that dream a reality by making a donation tomorrow, we would really appreciate it. Thanks again to Jennifer, Tayler, and Sarah for being amazing colleagues and scientists and community members. I really, really appreciate this. Thank you all. Have a good night.
Thanks. Thanks so much for your questions and thoughts too.
Thank you.
Thanks, everybody.